Okay, my goal from this talk is that you guys should leave terrified. um, but before I start, let me just quickly introduce myself. So I'm a research faculty at Yale using AI to translate geroscience. I previously built a computational biology startup. I'm really ingrained in this ecosystem. I've sort of patented a biomarker, sold it to a company. I work as a director of data science for a longevity clinic, and I also work with venture capital funds where I've invested quite heavily in the longevity space. But what I'm going to talk about today is something really interesting, are these virtual cell models. So what are these virtual cell models? How many of you have heard about a virtual cell model? Okay, quite a few people, yeah. So a virtual cell model is very cool. It can allow you to sort of do all of these things about testing molecules, testing potential therapies in a computer, right? But my thing is, are they cool enough? They aren't quite there yet where we can do this at mass in scale. They aren't quite ready where they're giving us the right results. And so we're doing new iterations of these models to improve them. And so one of the problems of these models is that these virtual cell models currently, or well, the first generation of them were using numbers, right, or not words. So they would typically, in the classical neural network format, they would take in these sort of gene expression values, right, put in these numbers and do some classification or regression or the likes of that. What that means is that you cannot feed in a lot of other kind of information. So there is a lot of information around papers, right? Like for the last 100 years, we've been talking about all of these gene interactions. There's so much information on the internet, right? We cannot feed it into these models. And we cannot ask these models questions, right? We cannot ask, why did this gene go up in response to this particular compound perturbation? We can't ask this model this question. So one of the things this really smart PhD student in collaboration with us and at the David Van Dyke lab did, he actually made gene expressions into words. So what he did was he essentially just rank ordered the genes. So the genes with the most expression essentially had the first rank, and the one with the least expression had the last rank. And these were pretty correlated with the count of the gene itself. And when you do this, you can actually feed this now into any natural language model like Grok, Llama, Gemma, even ChatGPT, right? But what's more interesting now, you can feed any kind of other natural language information, including papers, right? All of the publication literature data that you have, right? Perturbation information about compounds, disease, detailed disease information, tissue information, right? And this is really, really cool. But don't trust me. Trust the CEO of Google. We collaborated with them. This was a project that we did along with Google DeepMind. And we actually validated a bunch of different cancer therapeutics in silico and then tested them in wet lab and showed that these cancer therapeutics can be validated in a lab setting. So we tested in silico in a computer that whether they worked, and then actually showed that they worked in a wet lab setting. And sort of Sundar Pichai tweeted about this. But the problem is we cannot quite apply this to longevity very easily. There's some real issues here. Most of this data has not been trained. Most of this model have not been trained on sort of cell types. They include on sort of genetic conditions. Most of the perturbation data is only in cancer cell lines, which means it's not really useful for longevity. And most importantly, these models don't predict age. They don't predict biological age, which is most critical for doing longevity work. So we retrained this model, which is called cell-to-sentence, to predict longevity virtual cells, right? Trained on 10 million human cells. It does actually really good at predicting sort of chronological age or sort of biological age. And we've done this validation in a lot of different cases, but I just want to show one example. These are skin cells. Healthy ones are younger. Injured skin cells or from keloid, which is a type of disease, are actually older. But more interestingly, their predicted age when you actually put some different types of treatments like rapamycin, which is a type of very well-known longevity therapeutic. I'm just wrapping up. It goes lower. And OSKM, which is a type of epigenetic reprogramming, again, sort of decreases this age. But all of this is sort of wet lab tests. What I really want to talk about here, and this is my last slide here, is we now are able to do these in silico experiments where we can essentially say, okay, tell me which compounds are decreasing biological age across a bunch of different cells. And on the left-hand side are the compounds that on average decrease age across a bunch of different cells in the body. This is completely being done computationally on a computer. And these are well-known longevity compounds that decrease sort of age in the cell. Metformin, rapamycin, dasatinib. But on the right-hand side, what we discovered were compounds that are increasing biological age. These are well-known biotoxins: sarin, alpha-toxin B1, hydrogen cyanide. This to me is terrifying because the same model that is giving me well-known longevity compounds is also now predicting compounds that are bioweapons and biotoxins. I have not trained this model in any way to predict this. This model is not public yet, but when it does become public, anyone could potentially fine-tune this model to use it to develop bioweapons. And just FYI, this is one of many models out there that can do this. And so to people out there who are sitting here in the audience, all I want to say is, think about these bio-AI models that are out there that can be very easily fine-tuned to do very nefarious activities. Thank you so much.
Thank you. One question.
Yes.
What's your zombie apocalypse scenario for this?
Recently, you might have heard about sort of the attack by sort of Chinese state-authored, authorized attack on sort of US deployments using Anthropic systems. I think maybe in the next 5 years, we might see sort of bio-AI models being used by sort of state governments to develop model sort of even systems, microorganisms. I think it's a terrifying future. There are companies now that are being built to do this. There is actually a company called Red Queen Bio that's a spin out from MIT that just got $15 million to do this. So it's a very terrifying future where this is happening right now, and we need to think very hard about it. Yeah.
Red Queen Bio specifically was trying to do the opposite.
But
Like,
it's trying to do the opposite, but the argument, but they're essentially doing it, they're saying they're doing biosecurity, but effectively they're using these models to develop new compounds or sort of viruses that can protect.
got it.
Yeah. Yeah.
Well, thank you.